influences the acute inflammatory response, and how the ratios of macronutrients. So this will be the outline of what we're going to look at from the background up to the conclusions and future perspectives. Welcome to the talk. So apart from chemotherapy and radiotherapy, we know that nutrition is one of the interventions that are used in the management of cancer. Usually when we do nutrition management, we target to maintain the body weight of our patients and their lean mass, which is commonly lost because of cachexia, and also to support recovery and from therapy, which contributes with side effects. However, current research in nutrition shows dietary composition has the ability to modulate the immune system and also inflammatory pathways, and these have the potential of influencing cancer outcomes. So diets in cancer has a two-way pathway. One way, it is part of the supportive care for cancer, in addition to chemotherapy and radiation. But it's emerging that diet also has a very important role in modulating the immune system. So these are some of the diets that are currently being used in supportive therapy of cancer. We have high protein diets, which have been recommended in some studies, mainly to support lost muscle and to support the immunity, but also they help to modulate the inflammation and oxidative stress that is associated with some of the drugs that are used for cancer. We've heard about omega-3. Omega-3 is common from marine products like fish, not common from some of the oils we use like sunflower oils, which are rich in omega-6. So we have them being recommended to reduce inflammation associated with tumor necrosis factor alpha and interleukin-6. And also they are known to improve appetite. We have ketogenic diets, which are rich in lipids, and you limit carbohydrates. We do know tumors grow much faster than the blood supply. Okay, so at some point they don't have enough fuel and they, enough oxygen, and so they will prefer glucose as a source of energy, which can be metabolized anaerobically. So with ketogenic diet, the idea is to reduce glucose that is available for the tumor growth and provide lipids which requires oxygen, and hopefully they reduce tumor growth. However, there's mixed reactions to this kind of diets. However, we do know high lipid diets, they influence immune signaling, as we are going to see in a short while. Apart from those ones, we have different other diets. I will not focus on them, but you can see that are used in cancer. So what is the role of inflammation in cancer, which is my target? 25% of cancers are related to chronic inflammation. And inflammation as a process is not a bad thing, because once we have an offending agent, a microbe, a chemical, the body responds with inflammation. And normally the inflammation should facilitate the healing process. However, chronic inflammation can contribute, which continues for a long period of time, which is associated with chronic production of cytokines, can cause progression to tumor. And in diseases like diseases that are associated with chronic inflammation like diabetes, hypertension, we have increased incidences of cancer, and which is thought to be contributed by this chronic inflammation. Some of the cytokines that were of my interest, interleukin-1 beta, tumor necrosis factor alpha, and interleukin-6, have been shown to provide signals for growth and survival of early cancer through the nuclear factor, kappa beta, and STAT3 intracellular signaling pathways. And apart from just this, we have some interesting studies like we talked at the beginning about EGFR-related lung cancer. And a study here they found that once you block the interleukin-1 beta through interleukin-1 beta antibodies, actually you can lower the risk by 70% of a person who has started to show baseline inflammation that is related to EGFR. So clearly, modulating the cytokines can influence progression. Now, inflammation is not only important for the pathogenesis of cancer. We can see inflammation is there present in, we have therapy-induced inflammation. For example, we know tumor necrosis factor alpha, which is a good marker for inflammation, is increased when we have therapy with CTLA4 and anti-PD therapies. And we also have tumors that produce increased cytokines and contribute inflammation. And these ones are usually mediated by interleukin-6, where we have cytokine storms related to cancers, and this could be responsible for the cachexia, loss of weight that we see with patients with cancer. However, not all cytokines are bad. These cytokines I have described are pro-inflammatory cytokines. We have interleukin-10, which is an anti-inflammatory cytokine. Basically, it reduces the inflammatory response. And we've actually seen when interleukin-10 help having anti-tumor activity in renal cell carcinoma. So a quick question. If you are able to influence these cytokines, which ones would you want to increase theoretically, and which ones would you want to reduce? You'd want to increase interleukin-10, yes? And reduce the pro-inflammatory cytokines, interleukin-1 beta, TNF-alpha, and interleukin-6. I hope you all agree with me. Okay, so where does now diet come in place? So we do know macronutrients, we do know mainly we talk about them providing calories that we require for food. However, current research is showing they influence immune signaling. Good examples are like high lipid and high carbohydrate diets. We do know they promote oxidative stress by activating the pathways, nuclear factor kappa beta. Remember we saw it there with the cytokines? And influence and increasing cytokine release. And so they contribute to this chronic low-grade inflammation, which we have seen can be a good environment for the development of cancer, where we have likely to have DNA damage that causes tumor initiation and also progression. Then the inflammatory cytokines also are likely to promote when we have cancer there, they'll promote cell growth, angiogenesis, immune evasion. And although interleukin-10 we said is good, but because it brings down the immune system, is likely to facilitate the tumors to escape and contribute to metastasis. So what I'm saying is that we don't want excess of these cytokines, although they are good. We do not want an excess of them because they can facilitate growth of the cytokines. So when we have persistent inflammation, we have a good environment for DNA damage and tumor progression. So what was the gap for my study is that we have seen that diets do influence immune signaling. And although many studies we have seen, like the diets we have talked about that are in cancer, they focus either on reducing the calorie or reducing one particular nutrient at a time, like ketogenic increases the lipids. They don't really talk about what happens to the carbohydrates and what happens to the proteins. Previous studies have shown that the ratios of macronutrients in diet are able to influence aging, reproduction, and survival. However, how they play a role in the immune system, particularly the inflammatory cytokine response, is not very well established, although these epidemiological studies and association studies from what is seen in the clinical and the environment. So our objective for this particular was to see how varying dietary macronutrient ratios influence plasma cytokine responses, and we brought a challenge, a lipopolysaccharide. We know lipopolysaccharide is found on the gram-negative cell wall and is immunogenic. So we were sure that if we have the lipopolysaccharide, we'll actually see the body producing cytokines, and then we can gauge what is response in relation to the diet. And our model here was the Swiss albino mice. So what was our experimental design? We had male and female mice. For these particular studies, we used two, basing on the mind. Many studies use once either male or females, but we chose to have a combined study with male and female mice. We put them aged 6 to 8 weeks, and we fed them on one of 6 diets, having varied ratios of macronutrients. Don't worry about the abbreviation, I'll tell you what they are. So we have HCLP referring to high carbohydrates, low protein diet, high carbohydrate, low lipid diet. So you can see I'm talking about two macronutrients. The implication is the third one that is not mentioned is moderate. Okay, so for high carbohydrates and high lipid diets, the carbohydrates and lipids were as high as 75%, contributing 75% of the calories. However, in a previous study, which I'll not mention here, we found that when we put proteins as high as 75%, we found the animals dying. So we brought down the protein towards 60%. So for our high protein diets, the HPLL and the HPLC, the protein contributes only 60% of the macronutrient ratios. So again, in the previous work, we formulated these diets ourselves based on the American Institute of Nutrition 93M diet. We did a local formulation. You can see them there. We validated it and we found it's our local formulation. The control diet was actually much better than the locally based cereal diets that we use for mice. So we fed our animals on these diets for 15 weeks. And after 15 weeks, we classified them into two groups. A pre-LPS group where we injected them with 1 ml of normal saline intraperitoneally, and a lipopolysaccharide challenge group where we gave them 1 milligram of lipopolysaccharide intraperitoneally. And then we observed them for 3 hours. Then we sacrificed them and collected blood through cardiac puncture. And we isolated plasma from this sample and analyzed them using Luminex technology to assess the tumor necrosis factor alpha, interleukin 6, interleukin 1 beta, and interleukin 10. And then we analyzed this with the free software PAST 4.03 and Excel. So what did we find? So even without any immune challenge, we do find some of the cytokines, the pro-inflammatory cytokines, tumor necrosis factor alpha and interleukin 1 beta were actually produced in low amounts. The first two boxes, if we look at the graph of tumor necrosis factor alpha, show the high carbohydrates diets. In the middle, we have the high protein diets, and at the end, we have high lipid diets. So here, there's no challenge to the immune system. But note, the high lipid, low protein, and the high carbohydrate, low protein had significantly higher amounts of tumor necrosis factor produced even without an immune challenge. These values of tumor necrosis factor are within the normal range from previous studies, but for the high carbohydrate, low protein, and high lipid, low protein are above the normal ranges. We don't see such a big effect, a similar effect in interleukin 1 beta, although there's increased production. And then in interleukin 1, interleukin 10, the anti-inflammatory cytokine, we can see the low protein diet had significantly lower interleukin 10 production compared to the high carbohydrates or the high lipid diets. The ones that are showing BB, they are significantly higher than the high protein, low carbohydrate and the high protein, low lipid. After we challenge with lipopolysaccharide, now even interleukin 6 starts showing up. Remember, interleukin 6 is a pro-inflammatory cytokine only produced when we have infection. But again, look at the pattern. In TNF-alpha, the high protein diets, the cytokine TNF-alpha is lesser. Interleukin 1 beta also it's lesser. We are seeing also high protein, low lipid, producing lesser interleukin 6 compared to the other diets. And also interleukin 10, we can see the high protein diets having lower levels. If you look at the change, we went further to compare the cytokine levels, subtracting what we had after inducing and what we had before. I'd like you to note the graph of high protein, low lipid. It's consistently lower in all the cytokines, tumor necrosis factor alpha, interleukin 1 beta, interleukin 10, and also interleukin 6. So besides this, we went further and did multiple regression activities to check the interactive effects between the macronutrient ratios and the cytokine levels. And we used dietary protein and lipid as predictors. And what we found is that increasing protein intake significantly reduced the changes in TNF alpha, interleukin 6, and interleukin 1 beta. While increasing the lipid intake in diets increased TNF alpha but did not have effects on interleukin 6 and interleukin 1 beta. However, the interleukin 10 effect was unaffected by either macronutrients. Usually interleukin 10 is produced in response to the pro-inflammatory cytokine. So if TNF increases, the interleukin 10 will also increase to counterbalance the inflammatory response. So what do we conclude? If we look at our diets, we try to mimic them behind some of the diets we have talked before earlier that are being used for cancer. For example, our high lipid, low carbohydrate mimics a ketogenic diet. Our high carbohydrate, low protein diet mimics our cereal-based diet. If you think about what we eat in Africa, there's high carbohydrates and very little protein. And these carbohydrates from cereal-based diets. Our high protein, low carbohydrate diets mimics those diets that are adopted for either weight loss or athletic performance. When you want to build muscle, you want to have more protein. So we try to mimic our diets surrounding diets that are commonly used. And we have seen high carbohydrate and high lipid diets upregulate inflammation, as we have seen with increased TNF alpha, interleukin 1 beta, and interleukin 6. And they could do this through activation of intracellular pathways. Different studies have shown that high carbohydrates and high lipids, they have similar structures to the lipopolysaccharide. And lipopolysaccharide induces cytokine production through these pathways, nuclear factor kappa beta and AKT mTOR. And this is what we see, that people who have high carbohydrate and high lipid diets, they tend to have increased production of cytokines, which contribute to chronic inflammation. So we had also seen that when we had high diets, we had these cytokines being produced. Another mechanism we hypothesize could contribute to inflammation that we see with these diets is through oxidative stress and through the MyYD88 dependent mechanisms that can contribute to increased production of the cytokines. An interesting thing we mentioned is that we saw polyunsaturated fatty acids, the omega-3s, they have been used to modulate the immune system by lowering the inflammatory cytokines. However, in our study, we are seeing something different. However, our source of oil was soybean oil, which is rich in omega-6. So soybean oil, again, mimics our oils that we use to cook, sunflower oil and all that. Omega-3s are commonly found from marine products. Okay, so again, even as we're thinking about ketogenic diet, we have to think what is the source of fat, because omega-6 rich oils are likely to increase nuclear factor kappa beta activation and increase production of pro-inflammatory cytokines. For our high protein diets, we hypothesize they are likely to reduce nuclear factor kappa beta activity and hence reduce pro-inflammatory cytokine production. And the most interesting part of our study, which I think I forgot to mention, our diets were isocaloric. They were not the same caloric content, 3.85 kilocalories per gram. So our effects we are seeing, it does not mean that the person has to eat less. We just have to change the macronutrient in our diet. So that was one key studies. Remember, for our patients' diets, we don't want them to reduce the calorie intake. They need the strength, but we can modify what they are eating to give them the best outcome for the disease. So also our rapid activation of inflammatory cytokines after LPS challenge, we can see is that there's likely to be enhanced innate immune responsiveness in response to the different dietary composition. Remember the challenge, the cytokine production, we only did after 3 hours. Okay, in other studies, they need 24 hours after to see the changes in cytokine, but for us, in 3 hours, we are able to see it. So this rapid enhancement of immune system is good, because once an infection comes in, you want to clear it as fast. However, if it's persisted or exaggerated, as we have seen with the high carbohydrate and high lipid diets, could predispose to chronic inflammation. I think we saw this with COVID, where we had exaggerated immune responses in people who had diabetes and this could be some of the mechanisms. So what is the implication for cancer research? Come back home. We've seen chronic inflammation is important, not only as for initiation of cancer, but also it can modify it in cancer prevention and care. And so nutrition interventions have a potential to reduce the tumor-promoting cytokines and also help therapy tolerance and recovery, especially high-protein diets. But again, these studies were done in mice. We should proceed with caution before we directly translate them in humans. And so our study provides a basis for exploring these diets in cancer immunotherapy. So our conclusion is high protein diets reduce pre and post LPS cytokine levels in mice and suggest a protective immune modulating activity for dietary protein. And probably what we'd think about later is to extend this study to tumor-bearing models, like in mice. Probably want to assess what is the difference in protein. Here we used casein, but we want to see for us humans, we can be using plant-based or animal-based. Do they have the same effects? This will be a nice direction to take. And also to identify which particular amino acids could be involved. We saw some of the diets modify, were adding arginine specific amino acids, or which amino acids are responsible for this effect. So I would like to thank Charles in the lab. The work was done in the Makerere University Immunology Lab and my professors, Professor Chiwanuka and Dr. Kato. Some of the references, thank you.
Thank you, Helen, for the nice presentation. Yes, so just note your questions down. So the next speaker is Gift. And the Gift is also a PhD student at the University of Oxford. And he'll be telling us something about nuclear mechanotransduction. I'm really looking forward to know what that is. Just a moment to transfer his presentation.
Okay. All right, good morning, everyone. Thank you for coming through, and thank you, Helen, for a wonderful presentation. My name is Gift. I am at the University of Oxford in the O'Neill Lab under my expert boss, who's our keynote today. I hope I'm audible. Oh, Okay, so today I'll be talking a little bit more on the, thank you. Today I'll be talking a little bit more on the science, the fundamental biology, rather, a little bit step away from the very cool stuff we're discussing for the past few days. So I'll just briefly start on what is the extracellular matrix and introduce a little bit on this protein RASSF1A. There's so many proteins out there, but why a specific interest in that? And then just simply potentially linking the relationship between this RASSF1A and the extracellular matrix. But I would understand, as we have a lot of clinicians here as well, how do we translate these kinds of research? So if I could just zoom out a little bit, all the cells in the human body are exposed to some form of tension. This could be ECM stiffness, this could be pressure, tensile force, and this varies from cell to cell. And as you can see, as each cell is designed for specific responses, you'd find that they also adapt to various ranges of stiffness, where it's softer in the brain, and as you go towards the cartilage, it becomes stiffer and stiffer. But the interesting bit on that is there's a balance to this level of stiffness, the way the cells are responding, where too low of a stiffness is also associated with some form of disease. As you see, it can be linked with migraines. And in higher stiffness as well, it is also linked with some form of stiffness. And there needs to be a balance of this healthy stiffness. But when you look closely, you'd find as we go towards cancer malignancy, there's some form of increase in this stiffness. So if I could just zoom out a little bit and talk about the hallmarks of cancer. Hanahan and Wolbeck, they described, a lot of hallmarks of cancers, characteristics shared by different cancers. But if you look closely from, continuous replication, proliferative signaling, it all creates this chaotic microenvironment. And our particular interest is try to look into this stiffness, what we mean by this stiffness. So you'd find that when it becomes stiffer, a lot of studies have also indicated that it is associated with this tumor progression, angiogenesis, migration. It just creates this chaotic environment where the cells will start to proliferate and thrive. So my research, the one we're doing as well in the lab, is to even take a closer look at these cells. How do cells sense and respond to this tension? Why is it necessary? We've been talking a lot on genetics, we've been talking a lot on cytokines, which are very crucial in the space of cancer research. But also there's another dimension on stiffness, the biophysics on how cells respond to this. So if we look even closer to a cell, there is the nucleus, which houses the DNA. And our particular interests are around the nuclear envelope because it has been found to sense as well, to act as a mechanosensor. How is this changing in stiffness from the outside of the cell coming to the nucleus, which results in the DNA, adapting to respond to certain mechanisms? So when you look at the nuclear envelope, there's a lot of proteins, a lot is happening there. It is usually methylated or mutated in third of after TP53. And these are some of the publications from our lab, and they've indicated that, how RASSF1A plays in the space of suppressing tumor progression. So we're particularly interested on how this RASSF1A behaves. So just to take a step back, why we say RASSF1A is a tumor suppressor protein. When it is methylated, basically more of non-functional. I think Eric will talk a little bit more about that. It's a very interesting space. But you'd find that it is associated with a lot of these cancers. And if I am to introduce these graphs as well from one of the article here, where I would like to direct your attention is, if you see the lines where they are going down, it is simply showing survival falling in the cancers where RASSF1A is hypermethylated or basically not playing its role as a tumor suppressor protein. So I have two ways of explaining the slides. So on my right is the beautiful confocal microscopy images, and then on my left is sort of the brief, rough explanation of what we're trying to say. So you'd find that a recent, in our publication in the lab, we've seen that if you stretch the cells, if you induce this stretching mechanism on these cells, this RASSF1A protein, which we said is a crucial tumor suppressor protein, tends to move to the nuclear envelope, as you see with this nuclear ring localization. And it is usually on the inner nuclear membrane, because if you see on that beautiful graph, it has peaks which are indicative of the inner nuclear membrane. And it has also been found that this is actually happening in different cell lines. So as you see that when we induce tension, as cells experience this tension, this RASSF1A localizes here. Sorry. And in this recent study from our lab as well, they found that if we stretch the cells, we see that RASSF1A localizes to the nuclear envelope. We also see that ATR levels as well increase at the nuclear envelope. And this has also been, if we use hyposmotic tension, a different way of inducing this level of stiffness. And this is maintaining that. But as you see on the graph here at the bottom, we find that when we inhibit ATR is a DNA damage protein which is based in the nucleus, the RASSF1A level phosphorylation goes down. So if you can see from our diagram here, we see that ATR plays a role in activating this RASSF1A. But, sorry, a pop-up came up. But you'd find that the question still remains. How is this RASSF1A activation linked with the forces coming? So in some of the preliminary work from our lab, we've also seen that a stress response protein, a protein that becomes activated when the stress events happen, which is found in the cytoplasm, phospho-junk. You find that when you inhibit phospho-junk, the levels of RASSF1A activation goes down. And this has established sort of a potential link between stress, this protein phospho-junk, and potential activation at the nuclear envelope. And when we silence another player called GIP3, which is simply an interacting partner which binds and scaffolds phospho-junk, you also found that if we remove this GIP3, the RASSF1A does not become active. So why am I explaining this? We're simply trying to indicate how these players are playing a role in this RASSF1A activation. And what we then did in the lab was then question that, okay, so if these proteins are in the cytoplasm, where is the link with this RASSF1A activation? But the question we were asking here is, are they also recruited to the nuclear envelope, or is just happening within the cytoplasm? And we see a good recruitment towards the nuclear envelope when we induce tension, and which then indicates to us that this player GIP3 and phospho-junk could be involved with coming to the nuclear envelope and playing a role in activating this tumor suppressor protein, and in a way establishing this link. So we also tried a similar experiment in a different model, which was more in, putting cells in more collagen and try to experiment that from a more regulated stiffness level. And we see a similar trend as well, similar to our other collagen and stretching model. So this was more of a curiosity question, just taking a step outside. So if these proteins are coming to the inner nuclear membrane, how are they coming to the nuclear membrane? How do we question that? So there's a protein known as emrin, which is found at the nuclear envelope. Most studies indicate that emrin is more on the inner nuclear envelope. And our hypothesis is, could it be involved with flipping? So we needed to validate if this emrin is on the inside of the nucleus, or it can also be found on the outside. So what we did is we stained for lamin B1, which is in purple. You'd find that indicates the inside of the nucleus. And then we see emrin in green, that they also align nicely. But then we then did a cytoplasmic staining where we were not seeing anything in the nucleus, which is why you see there's nothing for lamin B. But then we still see some emrin on the staining here, which indicates to us that this emrin can also be found on the outside of the nucleus as well as on the inside. And there's a few studies which have also highlighted that. So it was pretty interesting because we're trying to understand if it is involved in bringing these molecules inside the nucleus when it comes down. And whilst we were on that journey, we were then questioning how can this apply in the context of, let's say, pancreatic cancer? So we added different cell lines. So the one etched, it's more of an epithelial cell line from a pancreas, and the MIA-PACA2 is a PDAC cell line. So we then did a Western blot to look for this activation of phospho-junk. And as you can see, the cancerous cell line, which is the HeLa, which is a cervical cancer cell line, the Eshtet, which is, let's look at it as the non-cancer cell line, and the MIA-PACA2, you see on the phospho-junk, they are only activated at 60 minutes of this tension, but we then questioned on the localization. We need to see the difference of this tension and stretching between the cancer cell line and the non-cancer approach. So what we then did is we then looked for this phospho-junk in the non-cancer cell line. And what we see is there's not really a significant change in this phospho-junk, which is in the cytoplasm, when we induce tension. But when we then look at the cytoplasm inside the nucleus, we also see that there's no significant change. So in this specific non-cancer cell line, there was no specific change on this recruitment. So out of curiosity, we then tried to look into this MIA-PACA2, the pancreatic cancer cell line. And what we see is the phospho-junk in the cytoplasm falls down. To us, it indicates that it is going somewhere. And then, as you might imagine, the next question is, can we look for it in the nucleus? And we also see that in the nucleus, it is going down. So where is this phospho-junk going? And there was indications as well in some of our preliminary studies that this phospho-junk is all coming to this nuclear envelope where it might potentially be playing a role in binding to this RASSF1A. So because we see that in this cancer cell line, there's this decrease in phospho-junk, it's going somewhere, we were then curious to look for an interacting partner that binds phospho-junk for this movement. So what we then did was blotted for a different protein known as GIP3, if you remember, which binds to phospho-junk for this movement. And we could not find it in the this non-cancer cell line. And as you can see in our Western blots as well, we couldn't find this GIP3. But then when we look for this MIA-PACA2, we do see this GIP3, and we think that it may be responsible in actually playing this role of bringing this phospho-junk, which was further validating some of our earlier findings. So I know I've talked a little bit too much on the science part, and the main point which I was just trying to put across is there's a relationship in cancer progression to the matrix stiffness. When the cells replicate, we see this chaotic microenvironment, a lot of stroma is released, and then this creates more and more tension, and also this drives this cancer progression. So we need to understand first the mechanisms of how cells tend to behave in this level of tension. And as we look close in these molecular pathways, we see that when we induce tension on the outer nuclear envelope, or when cells experience this tension, our hypothesis has been, how is this RASSF1A activated? Because a lot of work has been done on how RASSF1A prevents tumor progression. And we have sort of found a potential link in this, within this pathway. But what are the possible outcomes? What are the potential applications, you might ask? So, there is so many ways that this kind of research could be applied. But one of our particular interests is in the space of potential early diagnosis, right? Because we understand that 90% of all cancer-related deaths is due to late detection. And when we look closely, is if we could find ways of improving the way we detect these cancers, we could also contribute in preventing some of this cancer progression. So if we understand the mechanics behind it, could we actually try to detect stiffness on its own? Could we look for molecular signatures within the ECM so that we could integrate all these different approaches and mechanisms to try to detect this early. Because if you look at like pancreatic cancer, for example, the symptoms are difficult to detect earlier, unlike other kinds of cancers, right? So how else could we look at it from a different perspective? Because when we look at cytokines or elements that are released in pancreatic cancer, they are also sort of released at early stages. So how could we think different within this space? You know, we could even use different approaches from predictive computational modeling to look for these slight variations as well. So that is a little bit of the research we've been doing, and I would like to extend my appreciation to our lab, our supervisor, and the Rhodes Trust, our college, our lab, and everyone else involved in the research. Thank you.
Thank you, Gift. So our next speaker is Dr. Wanjiru Muridi. So just write down your questions. You'll ask them after this session. And I think Wanjiru is going to talk to us about